Everybody, welcome. My name is Mary Koshi with the Office for Diversity and Inclusion. Today's talk is on forming community ties to improve diversity in autism genomic research. We have Dr. Pilar Trelles with us today. She's an associate professor of psychiatry at the Icahn School of Medicine, and she wears several hats. She's also at the Seaver Autism Center for Research and Treatment. So this talk is one of many in a series to raise disability awareness throughout the health system. So we're really honored to have Dr. Trelles with us today to share some information about her research and her passion for improving diversity in this really important field. So I am going to stop my share. You may share your screen.
Okay. Can you see my screen? Yes.
Yes.
Okay. Do we know how many people do we have? I can't
see. We have about 21 so far, and people are joining as we get going. So yeah, feel free to.
I'll start. Okay. So I'm Pilar. I'm an assistant professor of psychiatry, maybe one day an associate professor. And as Mary mentioned, I share many hats. My interest really has been in bridging the work we do at the Seaver Autism Center, which in many ways is where I feel where we all think the field of mental health and autism is going, which is integrating findings from genomic research into everyday clinical practice. And with that say, I guess I'll share some of the work I've been doing over the past few years with the support from everyone here at the center. So I'll give, I know there's like a diverse audience here, so I thought I would give kind of a brief overview on genetic and genomic autism and why it's so important. And then I'll spend some time speaking about disparities in genomic research and trying to identify what have been the barriers to include more diverse populations. And then with that, I'll present the framework with kind of come up with to work with communities from diverse ancestry in a way that will help us improve recruitment and retention of ethnic minorities. And then I'm going to, I guess, open to questions and hopefully kind of wrap up the discussion in a positive note with everyone's suggestions and insight. So just to talk about genetics of autism spectrum disorder, just kind of a brief overview. So we know that autism is a condition as we speak today, we define it behaviorally by two sets of symptoms, which is social communication and interaction deficits and repetitive and restrictive behaviors. And then this is a very heterogeneous disorder, so it has a number of comorbidities. And what we think is that these different comorbidities really kind of share some sort of genetic or biological predisposition with them. The reason I mention this is that many times these comorbidities help us interpret some of the genetic findings we have and kind of guide the ideas that these will also guide treatment options. Okay. So in the past, we used to think of autism as just kind of one condition, and today we think of it as children that have, or individuals that have kind of different underlying biologies, and that really is highly correlated with the genetic makeup of those children, and that's why it's so important to identify what is going at that level to then understand what is the underlying biology of what is happening with those children. So for many years, we've known that ASD is a highly heritable disorder. We know this because we know that children, there's high recurrence in siblings. This is for kids that have a kind of all causes. So if there's a recurrence, we talk just about kind of classical ASD of about 11 to 12%, but then when we talk about children that have impairments in that domain, it can go up to 20%. We know that kids from monozygotic twins have higher concordance rate at dizygotics than siblings. And in earlier studies, we used to think that 50 to 60% was accounted for genetic causes, although now and more recently, I think we're setting that it's probably between 80 and 90%. This is a study from our group here at CBER that shows why the lower, it was thought earlier that there were lower rates, because at the beginning they were just accounting. So it was, in terms of time, if the study was done here, the second sibling had not been born, so then that family would have not been accounted for a degree of heritability. But then if the family is followed for a longer period of time, then you include the second sibling, and that increased the result for heritability. So just to give an overview, we know that there's multiple causes for that are underlying the genetic susceptibility to autism. One that is, this is a graph that I like very much because we have different types of genetic variations. We have rare, kind of common variations, and in general, these ones kind of convolute together and are more, and give the phenotype of autism. And they might interact with some environmental factors. And then that would either, for example, here, paternal age is considered a environmental factor, and that would create de novo mutations. So it's a very complex disorder, and although we understand much more about it, we're still kind of far away to fully understanding. However, it's very important to continue collecting information that will kind of help us understand the genetic variability of this condition. And this is very important. And what is happening today, and for example, what happens at the CBER Center is that, you know, understanding the genetics is kind of giving us molecular targets. We are growing, we're understanding what happens at an neuronal level. We create animal models, but then we also have, we are able to know, to identify therapeutic agents, and then that kind of can go into the children, and then you can create treatments that are more specific for the condition and are not very general. What happens today is that most of the treatments that we have for autism are, you know, very intensive behavioral interventions that are not as specific for a biological condition, but that are general for most of these kids. And kind of what, I mean, the way I see it is these children are so different from each other that you can adapt your behavioral intervention to what the kid has, but it's very difficult to adapt the molecule that would go for that kind of biological underpinning that particular child has. And behavioral interventions work, but they're highly expensive, they're very intensive, they require lots of hours, and our ability to change trajectories is not as great as we would want. So by understanding the biology, like it's happening in other areas of medicine, then we are able to actually offer better treatments. This is a graph that basically says the same, how gene discovery help us go into model systems in terms of animal models, and stem cells helps understand the pathophysiology, then drug development, and then we can bring that to the community. This is a graph that, if there's anyone here that works in other fields in medicine, this is obviously not unique. It's used, you know, it's highly used in oncologic treatment. And what is important is that you can think how the variance associated with race and ethnicity and environmental factors can have a huge impact in how any of these things interact with each other, and why it's so important to kind of move the field and try to collect information from different individuals. So as we move forward from, you know, kind of the importance of genetic and genomic research in our understanding in autism, I think I want to borrow some information from other fields that talk to us about disparities in genomic research and how that is today translating into disparities, or I guess widening the gap in terms of treatments. So I initially got interested in this. I grew up in Peru, and I spent a lot of my childhood at this hospital, which is very beautiful, where my dad was a doctor, and it was in one of the poorest neighborhoods in Lima. And it's always been that I think marked me a lot. And I've always been very focused on the contrast in which we live. And obviously, when you have, you know, disparities, this was, you know, another center was around there, but this was not unique to Peru. This happens here. This happens in New York City, where you have state-of-the-art centers, and then you have patients that cannot access, you know, services. So my initial interest when I went into this field was really to work and see how can we bring the services that we have to these communities in a way that they are more accessible. However, as I started seeing that, I started to realize that there was much more to it, and I started to see not only the now of the disparity in the treatment that many of the kids were having, but also just with an eye forward, how new advances were only focusing in a small population. And in many ways, it was not because researchers or the NIH do not realize how important it is to collect. You know, it's one of the focuses, you know, not just in mental health care, in all areas of science. It is that it's been very hard to reach some of those communities. So this is a sample that when I first started, this is a study published in 2009, and unfortunately nothing has changed much, where you can see here these are rates of diagnosis of you have white, it's, you know, kids from other ancestries get diagnosed. There's disparities not only in access to care, but also how, you know, how late they get diagnosed, and the amount of services they are getting. There's a study this year that was published by Constantino that found that kids from African American descent in the US, I think included thousands of samples. There were two very dramatic findings, and I saw it this morning, so I don't have a figure here, but one finding was that generally from the moment for, you know, white families that parents report a concern until they get help, generally 6 months to a year, and this is across the US. For African American families, it's above 3 years. And so that was, you know, that's very significant in the recent years. And there was no difference in terms of demographics. There was no difference in terms of maternal education, which I also found very concerning. And the other thing was that they realized that the rate of intellectual disability was higher. And generally kids that have intellectual disability, because they have more severe deficits, they get picked up earlier. But their concern was that it was not necessarily, they couldn't explain the differences in intellectual disability by anything else in the samples. And none of these kids had any of other risk factors for intellectual disability that they could explain. And they were concerned that some of these deficits were related to the fact that they didn't receive early intervention, because many of these kids were being diagnosed were above age 4. So that's still happening, and this is just something that is happening in kind of autism in general, let alone kind of moving to genetic research. In genomic research, which is the reason we are here today, and why it's so important that we kind of do something and think together in ways to not let the gap widen even more. This comes from large database, and you can see this is individuals that have participated in genomic research. Most of the samples come from individuals from European descent. This is basically the same. And these are world samples. And then there's you can see here, these are small groups. I accessed this on Friday. And what is happening is that this is translating into clinical practice, obviously, because we are learning less, or we know less about individuals from diverse ancestry. So this is something that we're learning from oncologic cancer. So this is the number of samples that you need to detect mutational frequency. and this translates into treatment. And as you can see, for white individuals, you know, the number of samples obviously changes depending on the type of cancer, but the samples that you need are, you know, they can get them. But then here, for the only ones that they could detect was for Black individuals for breast cancer. Everything else for any of the other races and ethnicities and for any of the other cancers, they had a deficit of samples, which is very concerning. And what they conclude, this is from 2016, is that despite the efforts that they are making to actually reach communities to try to diversify genetic research, there's still a huge gap, and that there's something that we need to do to improve this gap. This is another way to look at this. Obviously, with this, it can be translated into treatments. This is for hepatitis C treatment, and these are different genetic variants. Basically, what they say is for the individuals that are monozygotic, for this variant, they have a higher rate of response to treatment. But overall, because this is a variant that is much less common in Black individuals, they know that they are less likely to respond to treatment. And what they're saying is that, of course, this relates to genetic, and when, you know, the individuals that are higher for this variant, they respond better, but there's a much smaller sample. And part of the issue is that we just do not understand enough of the genetics of other populations in this group, like Hispanic or combined. It's different. So we are already starting to see and how this is impacting research in other areas of medicine. So as I was kind of doing this work and trying to engage families and work with families and trying to bring, kind of think of projects that would bring resources to the community or improve access to care, just of behavioral interventions. I started to learn more about genetics and genomics in general, and especially in autism. I started to see how it was impacting the field. And also it was very clear that not as many families from diverse ancestry were coming through our doors, and it had nothing to do with us not trying to engage them. I mean, it was a combination of less families were receiving the diagnosis that they needed. But there also was some sort of resistance from some families, I think, from coming in. This is another study. So these are guidelines from 2013, and actually it has not changed much, but it's kind of generally agreed that if you have a kid, you have a group of individuals with autism, and you do a chromosomal microarray, which is kind of the standard of care, about 10% will come back positive. If you add all of these different things, so Fragile X, different Rett syndrome, tuberous sclerosis, and then on top of that, you have kind of a comprehensive medical evaluation that will help you interpret some findings, you should be able to identify 30 to 40%, and probably this number has grown. I have another slide that looks at specific genetic panels, although that might be more complicated because it changes a lot from lab to lab. So there's this one study, there's very little that has been looked and seen. Okay, so this is what we know from kind of the research that we have. This is what we should be able to find, pulling data. And this is really what this group found. So they have a population, and this is much more reflective of what we have in the US. And they were expecting to find 30 to 40% of findings, and they only found 12%, which is pretty low. And all of these children had autism. A big number of them had intellectual disability and medical comorbidities. And then they have increased rates in certain variants, and they couldn't explain that, although we know that for tuberous sclerosis, there are certain groups that have higher rates, as is the same for Fragile X. And again, of course, they conclude that, you know, this is a field that is moving in the right direction, but we are still not at a place where, you know, we can fully interpret and serve everyone, and we need to make an concentrated effort to improve diversity in genomic research. So as we kind of move forward, one of the things that has been important has been to understand really what are the barriers to participate in research. There's, a wide interest in kind of trying to engage families. Unfortunately, there's been a lot of also missteps, I guess, from research in history. And maybe some of them have been ill-intentioned. Probably some of them could have been difficulties or relations with maybe cultural competency or how data was being interpreted. There's a group that collected data for individuals, for a community of Native Hawaiians, and they, everybody signed that they could keep their samples for future research, but I think it was not communicated well what it meant. And this ended up becoming into a lawsuit of years and years and years. And it's become, apparently, many of these communities really are very afraid or scared of participate of research because they felt they took their genetic samples and then they didn't use them in a way that was beneficial to them. It seems that they truly didn't understand the impact of what they were doing. So it's very important to kind of understand who you're working with and what are some of the things that are important to the community. There's also, poor knowledge translation. I think that at least for autism, the field is going in the right direction, but a lot of it maybe is not filtering enough into community providers and definitely not into families and why this is important. And as long as, the knowledge is not in the community, it's going to be very difficult to work with families for them to understand why this is important. And then, many of these places really just do not have the infrastructure or they don't have collaborative formworks. So if anyone has worked at a Medicaid clinic, you know that they're extremely busy, that you have to see a lot of patients. That is very hard. And, sometimes I think that providers are just trying to obtain for the patients the things that are kind of, everybody's just kind of trying to survive in a way. And you're providing the best care that you can with the resources that you have. And sometimes that do not give space to, speak about some of these things. And that is completely understandable. But the problem is that then that over time is just kind of widening the gap. As we were thinking about this, there's a big field of medicine, obviously, that is moving to form community partnerships where the research takes place in the community and has a direct benefit. And that is basically based on kind of forming a strong partnership where you really identify common goals with what they want. And then there's a clear and direct benefit to the community. I think anyone that works in research knows that, participating off research, even if it's beneficial for families, is very time consuming. You need to spend a lot of time and many times there's no like a direct clinical benefit. So if you are going to do something like collect genetic samples that might not have quote unquote a direct clinical benefit in a community that is, very tired and stretched thin, any sort of plan collaboration needs to really have a direct benefit to them. Because you don't want this to be a one-time thing. This needs to be kind of a sustainable model that will kind of last over time and from which we can all learn. So based on that, there's kind of 4 tenets. And I was very proud of myself when I thought of this. And then I went online and it was basically that someone else came up with something pretty similar, not that long ago. But anyway, so it's very important to have, obviously cultural competency is very important. You need to make sure that there's research dissemination and kind of building local capacity. If you do not build local capacity in the community that you're working with, this is obviously just going to be a one time, and there needs to be like a lot of trust and transparency. And this, obviously, a lot of this is key to research, to clinical research in general. But I think that when you're working with populations that have that feel that have been or that have a past or a bad history with science, and that today their just basic medical needs are not being met. I think all of this is significantly more important and needs to be at the center of what we do. So the first project that we started, we formed a partnership with one of our community centers, community clinics, a developmental disability center. And the idea was that we both were interested in improving disparities in autism research. I started seeing patients there at one of the clinics, which has made the work, in so many ways, I was there. So one of the things that I learned is, like, if you want to do community engagement, you really need to be at the community. You need to be working with the families and the parents and try to understand what are some of the things that are getting on the way. So this model, in so many ways, really helped us improve our capacity to recruit community samples. So this is before we started, and these are the numbers. This was the breakdown of what we had in terms of research participation. This kind of full disclosure includes 2 studies that were just targeting Hispanic and Black families, but those families also participated in all of the other research. So this is a breakdown of last year in terms of the patients that we had. And this is much more reflective of what we have in New York City. We're still, number of Hispanic families is basically very similar, as well as Asian, and what it's described as mixed. And then there's still like a shortage of African American families, but still is kind of an improvement. This is the Hispanic breakdown. So a number of Hispanic families identified as Black. Mixed ancestry mostly are families that are either Native American and white, or white and Black. And then there's a small number of families that identified as Native Indian, either from South or North America, mostly Mexico. With this project, we wrote a supplement to an R01 grant that we had here that was looking at electrophysiological markers in autism. And what we wanted was to use this framework for collaboration and see if we were going to increase our yield of recruiting sample from diverse ancestry. And we did. We had said, this is a very extensive kind of research battery, so we had said that we were going to add 30 families over 2 years, and we added much more. I think these are 45 families. Yes. Yes, these are 45. A couple of them actually were recruited outside of this kind of initiative, but most of them are. And so we kind of met our targets relatively quickly, which is generally not what happens in research, as I'm sure you know. And then me being Peruvian, of course, I thought, you know, this is kind of a great model. We should really try to take it, you know, outside. I also, you know, in some ways, I really wanted to find a way to collaborate with the center back home. So we did the same. So we started a collaboration with the Center on Sullivan, which is a center that's been in Peru since the late 70s and serves like 400 kids in Lima, and then more than 13,000 families around the country. And they have really now centers around the world. And it was very clear that, you know, the center, which is, you know, center a lot, and I'll talk a little bit more about, and I think that's why this has been so successful in family empowerment. And the idea was to increase access to care for underserved children with autism. So we started going in 2018. Alex Collison and I went a couple of times, and then I've gone after with a group of fellows. And what we did was direct kind of this idea of building local capacity. And that was through, so we saw a number of patients where we were there providing kind of expert consultation in mental health treatment. provided local supervision for local providers. And then we had kind of large seminars, and then I had small group discussions with parents and also some of the training staff at the center. We designed the study with their IRB. The families were involved from the beginning. They decided that they didn't want report of incidental findings, so they kind of made a decision, kind of a democratic decision. And again, I think they could do that. And in so many ways, this program for Expertise Center because it's centered in the idea that caregivers have empowerment in the care of their children. And what we did, part of the research design, was that we're not only going to collect samples for genetic analysis for research, but that we were going to provide families with clinical feedback, which we did. All the families agreed to storage their samples for future studies, but only related to neurodevelopmental disabilities. And there was always a clear push for wanting transparency. So we had, even before the study started, we had a Zoom with all the families that had agreed to participate, where they asked a lot of questions. And most of their concerns related to what will happen, you know, with the samples. How are they going to be stored? If I want to stop them, kind of remove them at some point, can we do it? If you have findings later, will they come back to us? And it went very smoothly. But again, I think it was built on this kind of strong partnership and this idea that there was a clear clinical and direct benefit to the center. So we started with a small pilot project that targeted 40 families with 45 programs. So some families had more than one affected child. All of the kids had autism and intellectual disability. We collected clinical information. So we did comprehensive psychiatric evaluations, collected some information on adaptive functioning, IQs performed by others when they were available, and then a number of behavioral measures. We did chromosomal microarray for all of the kids, and then if the chromosomal microarrays were negative, it was followed by a genetic panel. All the families received clinical feedback, and we partner with a clinical geneticist in Lima that helped us give genetic feedback to all of the families. And then all of the families participated of the Autism Sequencing Consortium, which is a large international consortium that was started by Joe Baxbaum at the Seaver Center. And all of the samples were sent for whole exome sequencing, and we are still waiting for the results. So this was a demographic of the kids. Most of the children were school-age kids. We had a few adults and some smaller children. And then the distribution, oh, sorry, this is a mistake. It's male, female. The distribution of the sample is what you would expect for a group of kids with autism. And then 100% of our sample had intellectual disability. 70% of them were nonverbal or minimally verbal. We calculated about 10% had mild intellectual disability, and 20% moderate intellectual disability. This comes from the few IQs that we had. It is an interesting program because it really centers on functional and daily living skills. So many of the kids that are nonverbal, they actually leave their homes and sometimes hold jobs. Sometimes some of them hold jobs. So their scores for adaptive functioning in those domains are pretty high, but probably their IQs, if they were measured, if we could with standard samples, would be significantly low. But they have a lot of functional skills, but that's just something about the program and how it's designed. And I guess that would be a completely different talk. About a third of them had medical comorbidities. A big chunk of those were epilepsy, and 2 of the kids had a history of leukemia in remission. This is interesting because, and I'll show you the results, one of the kids had a mutation associated both with autism and leukemia. And then the SEQs with social communication questionnaire, all of the participants had more than 20. The scores were much higher. This is the breakdown for the SRS, social responsiveness scales. All of these scores are very, very high. So these are kids that are very affected. I don't know why this one's so high. So it was a sample that was clearly very affected, that had intellectual disability, that had comorbid medical conditions. So we were expecting to find a high rate of positive genetic samples from the information that I presented. But we're very disappointed because we didn't. And this kind of highlights why it's important to learn more about this community, the individuals from diverse ancestry. So if you remember the study I presented earlier today, in the 2013 guidelines, Schaefer kind of concludes that at least 30 to 40% of individuals should have a positive rate. We had one of the samples we couldn't process. So we actually ended up processing 44 samples. So only one was positive for a chromosome microarray. And then we had a small positive that included between the CMA, no one had, Fragile X was not positive in anyone, but that was, I guess that was not surprising. And the gene panel that looked at 448 genes. All of these kids also had negative karyotypes, which I forgot to mention. So this is what you would expect, and we only got 7%. And the truth is that this is for individuals that are not as affected as the children that we evaluated. So we really were hoping to be able to provide them with more positive feedbacks. Having said that, the few families that got kind of clinical findings, it was interesting. There were a couple of adult kids that had been waiting all their life for kind of genetic confirmation. And it was, you know, at this point, both of these moms felt kind of very guilty that they had caused this to their children, and that might be cultural. And I think finding or understanding that had nothing, you know, there was a clear biological reason for the issues that their kids had, brought a lot of relief to the families. So this is the neurodevelopmental panel. We had, let me see here. We only had 5% positive rate. We had a really high number of findings of variants of unknown significant. Like on average, we had 4 per person, which is pretty high, and then no one had no findings. And probably most of these are benign findings, but because we don't have enough information about the population, they are harder to interpret. I found this study, it looks at exon sequencing in patients with autism, and about a quarter of them had positive findings. So this is much higher. Of course, this is a different panel, and this is exon sequencing. So we're expecting, and we'll hope to have our results, but still this is definitely not something that we're expecting. I think all of this kind of highlights the importance of doing work like this. The pilot study was kind of very successful. I mentioned that the school has 400 families and a number of families outside Lima. The study was left open for enrollment, and this year before the pandemic, we were going to go back and do kind of a similar work with 60 additional families that had already enrolled and signed consents for participation, but because of the pandemic, we had to cancel it. The center, as I mentioned, also works with they have hubs. Let me see if I can go back. In other areas of the world, and some of their centers have expressed interest also in participating. Of course, it's a much larger project, but I think this idea of working with communities that could benefit from the expertise that we have and where we can actually bring a clear benefit, where we can help to build capacities, where we can provide better care, and then educate and build knowledge in the families of the importance of doing this work and participating, even if it doesn't have a direct benefit to them, and kind of build this on strong on trust and transparency, seems to work very well and might be a way to kind of move this forward and bring what we are doing to other parts of the city and maybe the country and hopefully the world. So I think it's very important to continue doing this. Our findings highlight why it's very, very important that we make a concentrated effort to include people from all backgrounds. And I left enough time for questions if anyone has any. Thank you.